Hello students, welcome to the 8th class of Immunology. In this class, we are going to continue the discussion about innate immunity. This is the second part of the innate immunity discussion. And in this class, we are going to discuss about inflammation, innate lymphoid cells, complements and much more. Okay, so let's start the class and just check what we learned in our previous class. So in our previous discussion, we discussed the properties of innate immunity, what is innate immunity, how it works, then what are the different kinds of innate immunities or mechanisms which are present in our body. So we also discussed innate barriers, phagocytosis, opsonization, and PRRs and their types. Okay, so uh, we are going to continue the same topic, and in this class we will start with the inflammation. Okay, so what is inflammation? Inflammation, it is a complex biological response of the tissue to pathogens, damaged cells or irritants. Now what does it mean? That means it is a response, right? First of all, you should understand that inflammation is a response. That means it is against something, right? Now what is that which is against something? It is against the pathogens, damaged cells or irritants. When any of these three it enters into the body, then our body gives a response in the form of a complex event which we call it as a inflammation. Correct? So inflammation is a complex biological response of the tissues to pathogens, damaged cells or irritants. It is a protective mechanism to help eliminating the cause of injury, clearing out damaged cells and initiate tissue repair. Right? So it not only helps to eliminate the cause of injury, it not only clears out the damaged cells, but it also initiates tissue repair, which is very important. If our tissue will not get repaired, then there will be some problem in our body in that particular area, local area, and that will aggravate over the period of time. So our body would not like to do that. That's why inflammation will definitely, it will initiate the tissue repair, and once the tissue repair has started, then the tissue will heal and our body will function as a normal. Okay, now inflammation can be acute. So acute inflammation means it is a short term. That means it is just, it happened for a short period and it is crucial for healing and recovery. Okay, and then we have the chronic. Now chronic is also called as a long term, which is usually not resolved. Okay, it can lead to a variety of diseases including cancer, cardiovascular disorders and autoimmune conditions. So inflammation starts with a short term acute inflammation, and this acute inflammation is caused by a number of factors. For example, allergic reactions, frostbite, chemical irritants, infection, burn, trauma, cuts or laceration or stabbing. All these factors can cause acute inflammation, immediate for short term. But over the period of time, it becomes chronic inflammation if it is not get settled. And then this chronic inflammation leads to a number of disorders and diseases inside our body. And these include the cardiovascular diseases, rheumatoid arthritis, autoimmune diseases. Rheumatoid arthritis is a kind of autoimmune disease, then neurological diseases, and in some cases, cancer as well. Right? So, inflammation is important for our body against any irritant, bacteria, viral infection, or any injury. But it has to get resolved. That is the priority. If it doesn't get resolved, then the acute becomes chronic and then it leads to a number of disease. Clear? Now, there are hallmarks. That means there are certain symptoms or there are certain, characteristics of a localized inflammatory responses. We call them as a hallmarks. And these hallmarks are heat, redness, swelling, pain, and loss of function. So you can see all these five, they have lift up this inflammation. So they reflect increase in the vascular diameter. So we call it as a vasodilation, right? When the vascular diameter is increased, then we call it as a vasodilation. Now, when there is a vasodilation, there is a rise of blood volume in the area, correct? Because that correlates with that. If the blood vessel is narrow, there will be less blood. If the blood vessel is slightly dilated, then more blood. So that's why the blood volume in the area is increased. And when blood volume is increased, then heating of the tissue happens, because one of the functions of the blood is to maintain heat. And then because more blood is there, more RBCs are there, it also causes to become red. So we have the redness. All right. So all these things are related. Okay. Now, increased vascular permeability, it causes leakage of fluid from blood vessels and it causes edema. Whenever there is a leakage of body fluid in the local tissue, it leads to some sort of swelling, and that is called edema. Within a few hours, leukocytes also enter the tissue from the local blood vessels. So we have WBCs. Leukocytes are WBCs. By now you know this. So they also enter the tissue from the local blood vessels. So inflammation, it results from the activation of innate immune responses in the vicinity of the infection or wound. So vicinity of the infection or wound. So wherever there is the infection, wherever there is some injury, wherever there is some wound, then within the neighboring area, inflammation will be there. Clear? Yeah. Now, there are some resident tissue cells, for example, macrophages, mast cells, and dendritic cells. These are activated by PAMs, which are pathogen associated molecular patterns. You remember? If you don't, then go back to the earlier class. Or DAMs, which are damaged associated molecular patterns, to release chemokines, cytokines, and other soluble mediators into the vicinity of the infection or wound. So basically these resident tissue cells, why we call them as a resident tissue cells? Because they reside within the local tissue. They don't come from outside. They are present within the local tissue. So we call them as a resident tissue cells. And these resident tissue cells, macrophages, mast cells, and dendritic cells, they are activated by the PAMs or DAMs, and then they release the chemokines or cytokines and other soluble mediators into the vicinity of the infection or wound. So activated leukocytes phagocytose bacteria and debris and to amplify the response by producing additional mediators. So initially some response is there, but that response is increased or amplified, and that is done by some additional mediators. Now, resolution of the acute inflammatory response. Now, because inflammation has occurred, now it has to be resolved. So, resolution of the acute inflammatory response includes the clearance of invading pathogens, dead cells, and damaged tissue. So, first of all, the site has to be cleared. Anything, any invading pathogen or dead cells or the damaged tissue, if it is there, then it will be cleared. Then the activation of the systemic acute phase response. There is a systemic means which includes the whole body. So activation of the systemic acute phase response and additional physiological responses which include initiation of the wound healing. So wound healing also starts taking place in case of injury. The induction of adaptive immune responses, because this is innate response. So then now the next response is the adaptive response. So induction of the adaptive immune responses. But if it left unresolved, okay, then chronic inflammation onsets which causes more local tissue damage and possibly systemic effects. Systemic means which includes the whole body. So that means our acute inflammation has to be controlled, has to be immediately resolved. If it remains unresolved for long time, then it leads to chronic inflammation, and this chronic inflammation, it will onset more local tissue damage and possibly systemic effects. Now, if you can see in this picture, you will see how the inflammatory response initiates. So the tissue damage and bacteria, here you can see a tissue damage, and bacteria, you can see some bacteria here. These are the bacterial cells. So tissue damage and bacteria cause resident cells to release. These are the resident cells to release chemokines and vasoactive factors, triggering a local increase in blood flow and capillary permeability. So you can see this is a capillary, and these are the vascular endothelial cells. Now here these vascular endothelial cells are tightly adjoining to one another. There are tight junctions, but slowly you can see some gap is happening here. Can you see the gap is widening here, and this gap, it causes these cells which are present in the blood to migrate to the tissue. So exudate, complements, antimicrobial proteins as well as the cell. You can see here the cell is also going. So neutrophils and other phagocytes, they migrate to the site of inflammation, and that happens because of the chemotaxis, right? And then the phagocytes and antibacterial substances, they destroy the bacteria. All right, so that's how it happens. Now, there are some pro-inflammatory cytokines which are TNF alpha, IL-1 beta, IL-6, chemokines, prostaglandins and histamines, which are the part of chemokines. They act on local vascular endothelial cells. Okay, they act on local vascular endothelial cells to increase vascular permeability and the expression of cell adhesion molecules, which are CAMs, and chemokines like IL-8. So they increase the expression. Now cell adhesion molecules, they help to hold the cells which are flowing in the blood at that particular area, so that the cells can go to the tissue, they can migrate to the tissue. And chemokines like IL-8, they also causes a chemotaxic movement. Okay, the site is inflamed and exhibiting hallmarks. So just 5 hallmarks which you just discussed. So the site is inflamed and then it exhibits the hallmarks. Neutrophils are the first cells to reach the infection site, followed by monocytes that differentiate into macrophages. So the first responders are the neutrophils. And we have the systemic effects: TNF alpha, IL-1 beta, IL-6, upregulate COX-2, which activates prostaglandin E2, acting on the hypothalamus to cause fever. So whenever there is some infection, it always follows a fever. And why it happens? Because these cytokines, they upregulate this COX-2, which activates the prostaglandin E2, and this prostaglandin E2, it acts on the hypothalamus and it causes the fever. Now these 3 pro-inflammatory cytokines, that means TNF alpha, IL-1 beta and IL-6, they also act on the liver, inducing the acute phase response, which helps to resolve the inflammation. So it is important to act upon by these 3 pro-inflammatory cytokines on the liver, so that they can start the acute phase response, and this acute phase response will help to resolve the inflammation. Then inflammasomes. Now this is a new term which is coming up, so we will discuss in the next slide. Inflammasomes, they are responsible for the activation of inflammatory responses. So now the theory is that inflammation is activated, or inflammatory response is activated by inflammasomes. Okay, now if we see the inflammasomes, these are cytoplasmic multi-protein complexes in the activated immune cells. Okay, cytoplasmic multi-protein complexes, that means these are made up of different proteins and they are present in cytoplasm in activated immune cells, comprising, now what it's made up of: sensor protein, there is a sensor protein, there is an inflammatory caspase and or adapter protein which connects the two. So basically this is optional. Sensor protein and inflammatory caspases, they will be present in an inflammasome, and adapter may not be present. These detect pathogens and cellular stress triggering inflammation. And the fundamental processing unit for PAMP and DAMP sensing. So inflammasomes are the fundamental processing units. Remember, for pathogen associated molecular patterns and damage associated molecular patterns sensing, which actively participate in integration of their downstream immune signaling. Now, broadly classified, now these inflammasomes are classified into canonical. Canonical means which are standard and accepted, and non-canonical, which means they are slightly deviated from the standard pathways. So their pathways are not standard but deviated. They differ in activation mechanism, sensors, and biological outcomes. Okay, now we will see what is the difference between this canonical and non-canonical inflammasomes. So if you see the canonical and non-canonical inflammasomes and compare the features, you will find that the core sensor in canonical is NLRs, which is not like receptors, for example NLRP3 and AIM2 pyrin, and in case of non-canonical it is caspase 4, 5 and 11. The activators are, in case of canonical, PAMs, DAMs, crystals, and viruses, while in case of non-canonical, it is the activators are the cytosolic lipopolysaccharides, which are usually present in gram-negative bacteria. Now here, in case of canonical, the caspase is caspase 1, while in non-canonical, it is caspase 4, 5, and 11. Signaling is two-step, which is priming and trigger in case of canonical, while here in non-canonical it is single step, just LPS binding is enough. And cytokine processing, it directly cleaves pro IL-1 or IL-18, while this is indirect in non-canonical, it is via NLRP3 activation. So this NLRP3, everything we have discussed in our previous classes. The primary output in case of canonical is pyroptosis and cytokine release, while in case of non-canonical it is pyroptosis and it triggers NLRP3. The significance of canonical is there is a broad spectrum defense against pathogens, right? So canonical gives you a broad spectrum defense, while in case of non-canonical it is specialized for cytosolic LPS detection, which is critical for sepsis control. Now sepsis is when there is an infection inside your body. This is a different thing, we can discuss later. So it is critical for sepsis control. Now, non-canonical inflammasomes, they amplify canonical response. This is very important, right? So, non-canonical inflammasomes, they amplify the canonical response. And here the caspase 11 induced pyroptosis, it activates NLRP3 via potassium efflux, linking both pathways in defense against gram-negative bacteria. Clear? So here you can see how the structure of the canonical inflammasome and how the non-canonical inflammasome there. So let us understand the role of acute phase response in inflammatory response. So acute phase response is the changes in the concentration of many serum proteins during the acute phase of the disease. Now, when we call the disease phase as an acute phase, when it is at the peak, we call it as an acute phase of the disease. So, in that particular phase, there are changes in the concentration of many serum proteins. Okay. Now, acute phase response proteins, they change in response to inflammation. So, their levels go up and down as per the inflammatory reaction. The pro-inflammatory cytokines TNF alpha, IL-1 and IL-6, they act on liver to secrete APR proteins. So our liver secretes APR protein, remember. So liver is there to settle down the infection. It secretes the APR protein. Many acute phase response proteins are components of the complement system, contributing to innate and adaptive immune responses. So we are going to discuss this complement today only, but many APR proteins, they are the components of the complement system, or you can say that many complement components are also APR proteins, right, in both ways. Now, one protein is C-reactive protein. Okay, we call it as a CRP. And most, I don't know whether, but people usually, they are tested for CRP when their blood profiling is done to know the status of the inflammation. So the inflammation. Okay, so this C-reactive protein, it is an important liver APR and opsonin because it opsonizes as well. It's belonging to the pentraxins family. It bind to phosphocholine and carbohydrates on the pathogens. So here you can see there is an infection or trauma or tissue damage, and then there is an acute phase stimuli. IL-6 and IL-1, they are acting on the liver, and the liver is releasing the CRP, and the CRP is causing this bacterial phagocytosis, where not only as a complement but with the FCR receptor. So two other pentraxins, serum amyloid proteins and PTX, they also function as both opsonins and activators of the complement pathway. Persistent pathogens like TB, why we call them as a persistent pathogens? Tuberculosis, because they remain inside our body, right? Persistent pathogens like tuberculosis or other harmful substances, for example, monosodium urate crystals in case of gout. Yeah, gout is the disease of the joint where these monosodium urate crystals get accumulated. So we are using two examples. One is the persistent pathogen, which is the tuberculosis, and other is the these crystals, which are the monosodium urate crystals. They can cause chronic inflammation and continuing tissue damages and illness. Okay, so because continuing tissue damage because of the chronic inflammation is not resolved, they don't allow to resolve the inflammation, so inflammation keeps happening there, and that causes the damage and illness. Okay, now let us focus on the innate lymphoid cells. These are also the lymphoid cells, but they are used, or they are, they act in innate immune system. So they are developmentally related to T and B cells, but lack antigen specific receptors. Like in case of T cells, we have TCR. In case of B cells, we have BCR. But in case of innate lymphocyte cells or ILCs, we don't have any specific receptor for antigen. And they do not undergo clonal selection or expansion as these T and B cells undergo. These are tissue resident, generally present at the mucosal surfaces like intestine or lung, or in lymphoid and non-lymphoid organs. So they have a variety of places where you can find them. They play key role in early immune responses, tissue homeostasis and inflammation, particularly at mucosal barriers, which are the gut and the lung. So they provide very important immune response at these particular areas, at the mucosal barriers. ILCs are classified based on their cytokine profiles and transcription factors mirroring the TH cell subsets. Like we have the TH cell subsets. Likewise, we also have the ILCs, and we classify based on their cytokine profiles. So if you can see, we have three groups: group 1, group 2 and group 3. In group 1, the main subsets are natural killer cells and ILC1s. In group 2, we have ILC2s, and in group 3, we have ILC3s, LTI cells. These are the examples. Now, if we see the key cytokines, in case of group 1, we have IFN gamma and TNF alpha. And the transcription factors are TBAT, and their main functions and role is to defense against viruses, intracellular bacteria, cytotoxicity, particularly by natural killer cells. In group 2, the key cytokines are IL-4, IL-5, IL-9 and IL-13, and the transcription factor is GATA-3. And their functions is and role is to respond to helminths, which are worms, allergies and tissue repair. Okay, and in group 3, the key cytokines are IL-17, IL-22 and GM-CSF, and the transcription factor is ROR gamma T, and their function and role is mucosal barrier defense, lymphoid tissue development and response to extracellular bacteria and fungi. Okay, so these three groups, they constitute the innate lymphoid cells. Now, let us focus on natural killer cells, which are ILC1. So, they are the group 1 innate lymphoid cells, and they are the best known for their ability to rapidly identify and eliminate virus infected cells and cancerous cells. So, these natural killer cells, they have the immediate authority and capability to kill virus infected cells and cancerous cells. Their key functions is cytotoxicity for cells that lack MHC1 or display abnormal MHC1. Now you know that these MHC1 proteins, they are displayed in all nucleated cell or all nucleated cells of the body. So if there is some abnormal MHC1 or the cells are lacking MHC1, so that means the cells are abnormal and they immediately kill the cells. So this cytotoxicity is for cells that lack MHC1 or display abnormal MHC1. Then cytokine production, they secrete IFN gamma and TNF alpha, which coordinate and amplify the immune response, influence other immune cells and promote inflammation. That is their second function. Then immunoregulation, they interact with dendritic cells, macrophages, T cells and endothelial cells, shaping both innate and adaptive immune responses. So that is also their responsibility. And fourth is the immunosurveillance, which is essential for tumor immunosurveillance and for controlling viral infections. So they keep watching whether any cell is virus infected or whether any cell has become tumorous. Okay. Now, if we see the mechanisms of target recognition, then these cells, they use a balance of limited sets of activating and inhibitory receptors to determine whether to kill a cell or leave a cell. Limited set of activating receptors and inhibitory receptors. So that means these cells have inhibitory and activating receptors. So unlike B and T cells, natural killer cells are pre-programmed to respond immediately to appropriate stimuli. While B and T cells, they need the specific antigen, which is either bound with MHC1 or MHC2 molecules, or in case of B cells, just specific antigen is required, which can be determined by the BCR, and then they will expand. But in case of natural killer cells, these are just pre-programmed, they don't need that, they respond immediately, while in case of B and T cells, they need time. Okay. And to respond immediately to appropriate stimuli. Now, if you see receptors, we have activating receptor and inhibitory receptors. So, activating receptors examples are NKP30, NKP44, 46, NKG2D and CD16, and their ligands are stress-induced ligands, IgG, viral or bacterial proteins, and their function is trigger cytotoxicity and cytokine release. While inhibitory receptors include KIR, CD94 or NKG2A. Now their ligands are MHC or HLA1 and HLA-E, and they prevent attacks on the healthy cells. They also have the checkpoint, which is PD1 and PD ligand 1 on tumor or immune cells, and they suppress natural killer cells function in cancer, in case of cancer. So they suppress the natural killer function in cancer. So this is the PD1. It is present on the tumor or immune cells, which suppresses the natural killer cell function in cancer so that the cancer will not be attacked by the natural killer cells. Now, receiving an excess of activating signals than to inhibitory signals tells a natural killer cell about the abnormal target cell, and the natural killer cell is activated to kill. So, if more activating signals then compared to inhibitory signals, then it will be triggered to kill the cell. If more inhibitory signals are there and less activating signals are activated, then it will not touch the healthy cell. That means the cell is healthy, and that's how they determine whether to kill or whether not to kill. So NK cells can develop antigen specific and cytokine induced memory for enhanced effector functions, bridging innate and adaptive immunity. Now, this is pretty important point because you know that our memory cells are formed by the B cells and T cells. But here natural killer cells, they can also develop antigen specific and cytokine induced memory for enhanced effector functions, and this can bridge innate and adaptive immunity, because innate immunity, it doesn't have any memory function, but these cells have limited memory functions, and adaptive immunity of course has a well defined memory functions. So they act as a bridge between the innate and the adaptive immunity. Okay. Now, if we see the regulation of the innate response, we have the positive regulation, like we have PRR, PAMP, DAMP, trigger, signaling cascade. So you understand PRR, PAMP and PRR, DAMP? Don't? So PAMP is pathogen associated molecular pattern. Okay. And what is PRR? It is a receptor. Okay. And DAMP is what? Damage associated molecular patterns. So they trigger signaling cascade NF kappa B and MAP kinase, causing pro-inflammatory cytokines TNF alpha, IL-6 and IL-1 beta and chemokines production. Then pro-inflammatory cytokines and type 1 interferons, they boost immune cells recruitment and activation, inflammation, and shape the adaptive immunity. Innate immune cells like neutrophils, macrophages, dendritic cells and natural killer cells, they are activated to phagocytose pathogens. They release antimicrobial peptides and present antigens to adaptive immune cells. And their positive feedbacks are through cytokine signaling, which can further enhance immune cell recruitment and activation at the sites of infection. So these are the positive regulations. Now, in case of negative regulations, we have IL-10 and TGF beta, which suppress pro-inflammatory mediators and inhibit immune cell activation, promoting resolution of inflammation and tissue repair. Okay. Then we have the Treg cells, which modulate innate immune response by inhibiting the activity of other cells. Some TLRs, which are toll-like receptors, are localized within the endolysosomes. They reduce the likelihood of self-recognition and inappropriate activation because they are within the endolysosomes. So they are hidden. And then there are neural circuits which can modulate cytokine production and inflammation, providing another layer of control over the innate immune response. Okay. Now, there are certain pathogens which also evade these innate and inflammatory responses, and they have, beautifully developed some strategies. Now, let us see what kind of strategies they use. They hide PAMs, which is the pathogen associated molecular patterns, and it is done in case of most of the viruses and bacteria. Now, once they hide the PAMs, they avoid PRR detection, and therefore our response is very reduced. That is one way. Then they inhibit PRR signaling. For example, influenza, HIV and SARS COVID. The virus block or degrade PRRs or adapter proteins like MAVS, TRIF and MyD88. All of these are in the signaling pathway we have seen. And the effect is that suppressed interferon and cytokine response. right? Then interference with IFN pathway. So this is the example of hepatitis C, HIV and SARS-CoV. They block production of signaling of type 1 interferons and they degrade JAK-STAT components because JAK-STAT pathway is important. So they degrade the components. So impaired antiviral state and enhanced survival. That is the answer of this effect. And then blockade of antigen presentation. So herpes viruses and HIV are the examples which do this. They inhibit MHC class 1 and class 2 expression or function, and therefore there is decreased natural killer and T cell recognition. So these are four evasion mechanisms. Now next is the evasion of natural killer cell activity, and this is shown by cytomegalovirus and HIV. So how do they do? They express MHC like molecule, down-regulate stress ligands to inhibit natural killer activation. So basically they are inhibiting the natural killer activation. So they escape from natural killer cell mediated killing. Then another strategy is that modulation of cytokine or chemokine networks, and it is exhibited by pox viruses and herpes viruses. So they encode certain viral cytokines or chemokines which mimics or inhibitors. They altered the immune cells and recruitment and activation. That's how the effect on host cell. Then another strategy is the inhibition of the apoptosis done by or shown by many viruses. So the mechanism is simple. They express anti-apoptotic proteins to prolong infected cell survival, and therefore the infection remains persistent, because if the cell will be died, then the virus will not be able to replicate. But here they prolong the anti-apoptotic they prolong the infected cell survival by expressing the anti-apoptotic proteins. And then another strategy is the breaching physical barriers in case of influenza, HIV and flaviviruses. They disrupt epithelial junctions or exploit permissive cells at barrier sites, and they facilitate entry and spread. Okay. Now, how innate and adaptive immunity collaborates, this diagram we have discussed in our earlier class also, but here it is also important, so that's why I have put it here again. So you can see here the red color pathogens, these are picked up by the phagocytic cells which are yellow. And in case of innate stage, we have innate immune cells like macrophages, dendritic cell, mast cells, neutrophils and monocytes, which are activated through PRR or PAM, and then antimicrobial and pro-inflammatory responses are given. Phagocytes and these dendritic cells, they carry pathogen. So you can see here they are carrying the pathogen pieces to nearby lymph nodes to help activate adaptive immunity. So here you can see this, then the cell is taking here to the lymph node, and in the lymph node you have the T cells and the B cells. So T and B cells, they are clonally selected and beginning the adaptive response. So now from the innate stage it has moved to adaptive stage, and T and B cells, they team up and continue to encounter antigens in the lymph node. So here it will antigen, and then they are replicating as well as they are producing the memory cells, and they are also migrating. Okay, so B cells secrete antibodies and memory T and B cells are generated. So that's how the innate and adaptive immunity, they collaborate. Now let us see the interaction between the innate and adaptive immunity. So PAM's binding, which is to the disease PRR, okay, dendritic cells PRR, induces maturation, and we call it as a DC licensing. So here it is important to note that the DCs, they are not usually they express the type 2 MHC molecules because they are the professional antigen presenting cells, and then they present the antigen to the T cells. That is the normal routine, but here they are using not the MHC associated mechanism, but they are showing the PAMs, and this PAM binding to DC's PRRs, because DCs also express PRRs, and PAMs bind to DC's PRRs, they induce the maturation, and this we call it as a DC licensing. Now they express more M.Sc class 2 and co-stimulatory proteins, which is CD80 and CD86, which is recognized by the T helper cells. So our T helper cells, they recognize MHC class 2 molecules which are with the antigen as well as the co-stimulatory proteins CD80 and CD86. DCs also secrete specific cytokines and influencing the activation of the suitable T helper subsets. So because T helper has a lot of subsets, so which subset has to be activated, it is determined by the cytokine which is secreted by the DCs in this response. Now if we see the differential signaling through DCPRRs, so you can see here that tissue bacteria and endosomal foreign nucleic acids induce IL-12 secretion to differentiate T cell into Th1 subset. So you can see here this is the bacteria, okay, and it is producing IL-12, and this IL-12, it is differentiating the native cell into Th1 subset, and Th1 secrete IFN gamma to activate macrophages and natural killer cells to kill pathogens and infected cells. Now PAMs from worms, you can see here this is the helminth worm. Some bacteria and fungi induce disease to secrete IL-10. So here you can see they are secreting the IL-10, which helps along IL-4 and IL-13, okay, from nearby basophils and ILC2 to differentiate T cell into Th2 subset. So here you can see IL-4, IL-5, IL-13, and they are differentiating into Th2 subset. Fungal PAMs binding T cell differentiation to Th17 cells. Okay, so here you can see this is the fungal cells, and when it is showing up here, it is differentiating the native cells into Th17 cells. And TLR26 induces DCs. Here you can see TLR2 and TLR6. This is TLR2, TLR6. They induce DCs to convert the vitamin A into retinoic acid and to produce cytokines like IL-10, TGF beta. Okay, so here you can see this is IL-2. Yeah, here you can see IL-10, RA, and TGF beta. RA is retinoic acid. All these jointly induce Treg formation, which inhibit immune response. So you can see here it is, differentiating into T regulatory cell, and that inhibits the immune response. So that's how there are differential signaling through the dendritic cell PRRs. So there are so many PRRs, which PRR is activated and what kind of cytokines it is producing and what kind of subset it is going to differentiate. It is helping to differentiate in T cells. So that is important, and that's how it works depending on what kind of the activators or ligands are there. One way to develop tolerance to food substance and beneficial microbiota, because our gut has a complete microbiome which is very helpful in our digestion, as well as we tolerate the foods, because foods are also the foreign material which we ingest daily. So that has to be tolerated, and that's why these Treg formations, they are very important for to develop a tolerance. Okay, now the next is innate immune sensing in B cells. So here you can see this is the B lymphocytes. So B lymphocytes, they are basically they have the antibody molecules, and then they bind with the antigen. That is their adaptive response. But B cells also have the innate immune sensing capability, and this innate immune sensing capability comes with the toll like receptors and other receptor molecules. So here you can see TLR2, it causes decrease in B cell maturation and increase in chemokines and cytokines, while TLR4 interaction increase B cell maturation. Now there are endosomal TLRs, TLR3, TLR7, TLR9. So TLR3, it increases the pro-inflammatory cytokines. TLR7 increase the pro-inflammatory cytokine, chemokine, spontaneous germinal center formation, increase in autoreactive antibodies, and increase in hypergamma globulinemia. And in case of TLR9, it causes increase in antigen presentation, increase in sensitivity to BCR stimulation, increase in survival and proliferation, increase in pro-inflammatory cytokines, increase in chemokines, increase in spontaneous germinal center formation, and increase in autoreactive antibodies and hypergamma globulinemia. Now there are other receptors also like C gas and sting. So C gas cause decrease in parasite burden and decrease germinal center response, while sting causes increase in disease related mortality, disease related pathology, increase in cytokine production, decrease in JAK-STAT1 activation, and decrease in antibody response. While RIG1 through NAVS, it causes increase in disease related pathology, which is lupus, increase in cytokines, increase in spontaneous germinal center formation, increase in autoreactive germinal center formation, increase in autoreactive antibodies, and increase in TLR3 and TLR7 expression. So here you can see basically that how B lymphocytes, using their innate receptors, how they can also respond, although their main function is to provide immunity using the antigen, and BCRs are spread all over their surface, but these small receptors, they still innate receptors, they can also activate the B lymphocytes in different manner. Now if we see the same thing in T lymphocytes, so TLR2, it is inhibitory Treg function, it decrease in B fragilis model and in Caenorhabdita albicans model. Caenorhabditis albicans model, and they increase in Th17 differentiation cytokines, increase anti-apoptotic molecules, increase in Treg proliferation and survival, decrease in Th1 response, and decrease in chemotaxis. In TLR4, they increase in Th17 differentiation and cytokines, they decrease Th1 differentiation, they increase the adherence and chemotaxis. While the endosomal TLRs 3, 7 and 9, you can see here the 3 increases the anti-apoptotic molecules and survival, while in case of TLR7, decrease in Th17 differentiation, decrease in proliferation and energy, and increase in Th1 cell death. While in case of RIG1 and LPG2, it increases the survival and decrease the death receptor signaling, while RIG1, it decrease the proliferation and increase the apoptosis. Okay, and sting increases apoptosis, increasing ER stress. ER stress is basically when there is an accumulation of misfolded or unfolded proteins in the endoplasmic reticulum. We call it as a ER stress. So it increases the ER stress and decrease the proliferation. And through NLRC, if we see, this is Th1 proliferation, decreased Th17 proliferation, and decrease in the cell metabolism. Okay, so this is just the summary slides in case in both B and T lymphocytes, and how do they implement the innate strategy when they are in circulation. Now let us come to the another component of the innate immunity, which is the complement system. So complement system is a network of secreted or surface expressed proteins, and they increase the body's ability to eliminate foreign invaders, damaged cells, and immune complexes through a cascade of reactions. There are more than 50 proteins as of now currently, and they act as effectors, receptors, and regulators, and the system bridges innate and adaptive immunity. And if we see the key points, the protein precursors are synthesized mostly by the liver and keep circulating in inactive form. Okay, then there is a cascade mechanism which is triggered by pathogens or antibodies, and then activating proteins sequentially by cleavage or change in conformation, they are activated. So key triggers are PAMs, antibodies, or damaged host cells, and the proteases are synthesized as inactive proenzymes or zymogens, which become enzymatically active only after proteolytic cleavage, usually by another complement protein. Okay, so basically you understand that complement is a system of proteins, and there are currently more than 50 proteins, and you can categorize them in effectors, receptors and regulators, and they bridge innate and adaptive immunity, because they are the some once there are three systems which we will see, which are the part of the innate as well as the adaptive system, and they are the proteins are secreted or they are synthesized by liver, and these proteins are inactive initially, but then they are activated by their cleavage or by the change in conformation, and when they are triggered, and usually the triggers are PAMs, antibodies or damaged host cells, then they cause the proteases to be synthesized in inactive form, and then the proteases are activated by other complement proteins, and that's how we will see in the network. Now, if we see the complement, so complement components can be classified into 7 functional categories. Okay, so all the complement components, they can be classified into 7 functional categories. First are the initiators, and examples are C1q, MBL, and phycolins. So these initiators, they initiate the pathways by binding to pathogens, damaged cells, or immune complexes. So they are initiators. Then we have the convertase activators. For example, C1R, C1S, C4B, C2A. And then we have enzymatic mediators like C3 and C5 convertase. So these activate convertases and mediators of the pathway. So basically their function is to activate the convertases and mediators of the pathway. Then we have the opsonins. They opsonize targets for phagocytosis. Then we have anaphylotoxins, which induce inflammation. Then we have the membrane attack complex, which punch holes to lyse the membrane. And then we have the complement receptors, which are present on phagocytic cells, granulocytes or erythrocytes, right? And then we have the regulatory proteins, which prevent unintended injury. So they are regulatory proteins. So that's how complement components are classified into 7 functional categories. Okay. Now, if we see the complement activation pathways, we have 3 pathways. One is the classical pathway, another is the lectin pathway, and third one is the alternative pathway. Now, classical pathway is basically antigen antibody immune complex mediated or triggered. Lectin pathway through PAMP recognition by lectins, and alternative pathway is spontaneous hydrolysis or pathogenic surfaces. Now, there are some key similarities among all these 3 pathways. All converge at the activation of C3 and the formation of C3 convertase, and later steps that resulting in opsonization, inflammation and lysis. So if you can see, all these 3 pathways, they converge here at the C3 convertase, C5 convertase and the termination. So all 3 have the common mechanisms here and the common outcomes here, right? First thing. Second, all are tightly regulated to prevent host tissue damage, correct? Then we have key differences also. The key difference comes that classical is Ig dependent and it links to adaptive branch. You can see here, this is antibody dependent, which is the classical pathway, and because it is antibody dependent, then it is linked to the adaptive branch, while the others are Ig independent. Here there is no antibody, here are we have the carbohydrates, and here we have the pathogenic surface proteins, and they are in innate. So the the initiator molecules and the pre C3 events, they are also the key difference. So you can see here the initiator here is different. Here we have the antibody, here we have the carbohydrates, and here we have the spontaneous hydrolysis or pathogenic surface. So that is a different. And the pre C3 event, this is C3 convertase event, and pre C3 events are these events in the yellow part. So all these 3 are different. Okay. Now, if we see the 3 different pathways, classical, lectin and alternate, and if we compare them, the initiation or trigger in case of classical pathway is binding to the antigen IgG or IgM complex. In case of alternative pathway, it is the direct activation by microbial surfaces. And in case of lactin pathway, it is the binding ambial to mannose on microbes. The key initiators are, in case of classical, it is C1 complex, which is C1q, C1r, and C1s. In case of alternative pathway, it is spontaneous hydrolysis of C3, factor B, factor D, and properdin. And in case of lactin pathway, it is MBL and MBL associated serine proteases, which are MASP 1 and 2. Antibodies are required in case of classical pathway, but not in case of alternative and lactin pathway. In case of classical pathway, they have the role in adaptive immunity, while alternative pathway and lactin pathway have the role in innate immunity. C3 convertase in case of classical pathway is C4b2A. In case of alternative pathway, it is C3b, capital BB, and C4b2A in case of lactin pathway. C5 convertase is C4b2A3B, and in case of alternative pathway, it is C3bbb3B, and in case of lactin pathway, it is C4b2A3B. It is important to note these differences because they are asked in exams, and usually you should understand that what are the components which are involved in C3 and C5 convertase formation. The unique activators are antigen antibody complexes, apoptotic necrotic cells, and acute phase proteins in case of classical pathway, while bacterial endotoxins, IgA, IgD, cobra venom factors, and nephritic factors in case of alternative pathway. And then in case of lactin pathway, it is the mannose and N-acetyl glucosamines or microbial surfaces. So here you can ask me that why, if IgA and IgD is there, then why we are not putting it into the adaptive immunity, because these are the unique activators and these are not the usual activators. Okay, that's why we have put it into the innate part. Although you can say that yes, it can it can act both in innate as well as the adaptive. If we see the regulation, it is controlled by C1 inhibitor and others, while in case of alternative pathway, it is regulated by factor HI and properdin, and in case of lactin pathway, it is similar to classical after C4 and C2 activation. So that becomes same. The phylogenetic age is evolutionarily newer. So classical pathway is slightly, it has developed later, while the most ancient is the alternative pathway, and lactin pathway is similar to classical but part of the innate system. Amplification, if we see, there is no direct amplification, and alternative pathway is through C3. Yes, there is an activation and amplification, and again lactin pathway, just like classical pathway, there is no direct amplification. Although it might look very boring slide, because we have to compare all these three, but if you are preparing for the examination, then this slide will give you the entire gist of all these three pathways, their key differences, and the key activators and their convertases and their regulatory pathways and the amplification and phylogenetic age. Okay. Now, if we see the nomenclature of the complements, the first proteins discovered belong to the classical pathway and are designated letter C. Because C is classical, so they are always designated as letter C, the first proteins which were discovered, followed by a number in order of their discovery rather than the sequence of reaction. It is important to note that the sequence of reaction was different, but because they were discovered one after another, so their numbers were in the order of their discovery, not the sequence of their reaction. So products of cleavage reactions are designated by adding a small letter as a suffix. So cleavage of C3, it causes C3A, which is smaller, and C3B, which is the larger unit. So you can see here we have added A and B in small case. By convention, the larger fragments is designated by the suffix B. It is now again important to note that it is not A but it is B, which denotes the larger fragment, with the exception for C2. We have exception here in where the C2A is larger fragment. So you have to remember this thing, that C2A is larger, but usually in other cases it is B suffix which denotes the larger fragment. Another exception is the naming of the C1Q, C1R and C1S. They are not the cleavage products, but we write as a, small cases here, Q, R and S. But they are not the cleavage product of C1, but they are the distinct proteins and together comprise C1. So basically they are the part of the C1, three components, but they are not the cleavage product. Okay, they are the distinct proteins. The proteins of the alternative pathway are designated by different capital letters. For example, we have factor B, this is B is capital, factor D, D is capital. So whenever there we have the capital letters, that means they are the alternative pathways. And their cleavage products are added in lowercase a and B. Thus, the large fragment of B is called capital B small b and the small fragment is called capital B small a. Okay. Sometimes activated components are designated by a horizontal line above them. So C2A, and there is a line here. So whenever there is such line, that means they are the activated components. All right. So here this is a table, and just you can look at it by pausing the video and you can see, what are the different components and, how their functionality is related to them. Okay. Now we see the classical pathway, lectin pathway and alternate pathway. So let us compare the classical and lectin pathway. So here the initiation is the IgG or IgM which binds to antigen. So you can see here the red antigen and the green antibodies, they are bind here. And then C1q, this is C1q. Okay. This C1q, it binds to antibodies. So you can see here it is binding to the antibody here, and C1R and C1S get activated. So because C1R and C1S are present, it is a part of the C1 complex, so they get activated by binding. Now C1 cleaves C4 into C4A and C4B. Okay, and sorry, C2 into C2A and C2B, and they form C3 convertase C4B to A. So here you can see C4B to A. This is the C3 convertase. Clear? So here this C1S cleaves that. Now in case of lectin pathway, we have the mannose binding lectin or phycolins which bind to carbohydrates on microbial surface. So here you can see this ambial or phycolins, they are binding to the carbohydrates. This is the microbial surface and they are binding here. Ambial associated serine proteases MASP1 and 2 cleave. So here you can see this is the MASPs, this sky blue color component here, and they cleave the C4 into C4A plus B and C2 into C2A plus C2B, just like in case of the classical pathway which we discussed, and they form C3 convertase. Okay, so here you can see the same pathway, the lectin and classical pathway, they are going through the same manner. Correct? Now C3 convertase cleaves C3 into C3A and C3B. All right, and C3B helps to form C5 convertase, which is C4B to A3B. So here you can see it is C4B to A3B. So this is C5 convertase. Now C5 convertase cleaves C5 into C5A and C5B. So here you can see C5B, C5A and C5B, and C5A causes inflammation and C5B causes lysis. Correct? Now C5B binds to C6, C7, C8, C9 to form membrane attack complex, and C5B to 9, they form a membrane attack complex and they cause lysis. Correct? Now in case of alternative pathway, it is initiated in three ways. Now first is the tick over pathway. It uses the 4 serum components C3, factor B, factor D and properdin or factor P. Now C3 is constantly made and naturally inactivated by properdin. Second is by properdin, and third is by proteases such as thrombin and kallikrein. So that's how in three ways this pathway is initiated. Now in case of tick over pathway, there is a spontaneous C3 hydrolysis which is C3H2O and binding to factor B cleaved by factor D. Okay, so it form a fluid phase C3 convertase which is C3H2 OBB. So here you can see this is the fluidic phase of the C3 convertase, which cleaves C3 into C3A and C3B. So here you can see it cleaves the C3 into C3A and C3B. Now C3b, it binds to pathogen's membrane, it binds with factor B cleaved by factor D, forming C3 small b capital B small b, is stabilized by properdin, which is factor P. So here you can see, right? And addition of a second C3b to C3bbb complex forms C5 convertase. So here you can see C3bb, C3b, this is C5 convertase of the alternate pathway, and it is stabilized by properdin. C5 convertase cleaves C5 into C5a and C5b as we discussed, and then C5b binds to C6, C7, C8, C9 to form membrane attack complex, and this membrane attack complex, it causes the lysis. Now properdin, in case of the second initiator, the properdin, it first binds to the microbial membrane and stabilizes C3bbb complex. Okay, so this complex, it stabilizes. And in case of thrombin, it cleaves the C3 and C5, releasing C3A and C5A. So they also can activate the alternate pathway. Now, if we see how the membrane attack complex is generated, so you can see the newly formed C5b, this is C5b, it reacts with C6, this is C6, to form a stable complex which is C5b6 complex. So this is C5b6 complex, right? And binding of C7, now this C7, it binds here, it results in a hydrophobic complex that targets the membrane. Now we can see here, this is the MC5B7. So it targets the membrane because membrane is hydrophobic. It is binding here, C7 is binding here, and then it is targeting this membrane. Membrane insertion is initiated upon binding of C8. So you can see here, this is the C8, and now C8 is also bound here, and then this C8 has started the insertion. So slowly this complex is inserting, and then 12 to 18 copies of C9. So you can see here C9, poly C9, they polymerize to form the pore forming ring structure. So you see here that they have started forming a ring structure. So this forms a membrane attack complex. Now this pore induces the lysis of microbial membrane because there is an inrushing fluid and it can go inside and then it can just kill the microbe. Now some M.Sc targeted cells, they undergo apoptosis but different from regular apoptosis. So these are termed as apoptotic necrosis. Alright. There are certain M.Sc targeted cells, membrane associated complex targeted cells, which undergo apoptosis, but this apoptosis is different from regular apoptosis. So this we call it as a apoptotic necrosis. So if somebody asks you what is apoptotic necrosis, so you can say that apoptotic necrosis is slightly different from regular apoptosis where the M.Sc targeted cells undergo apoptosis. Now M.Scs can be removed from the cell surface either by shedding M.Sc containing membrane vesicles into the extracellular fluid or by internalizing and degrading the M.Sc containing vesicles in the intracellular lysosomes. So that means if the cell has got the M.Sc complex, still the cell can save itself by two ways. Either it will remove the M.Scs and it will just throw it into the extracellular fluid, or it will internalize the vesicle through and it will degrade in the lysosome. If a M.Sc is shed or internalized soon enough after its initial expression on the membrane, the cell can repair any membrane damage and restore its osmotic stability. So as soon as the membrane attack complex is formed and the cell is able to either throw it out or internalize and degrade it, then it can repair the membrane and it can maintain the osmotic stability and the cell will not die. In gram-positive bacteria, the complement proteins cannot penetrate the cell wall because it's thick cell wall. While in case of gram-negative bacteria, complement first permeabilizes the outer layer and following restriction of the thin cell wall, it lyses the inner bacterial membrane. So remember the difference between the complement activity upon the gram-positive bacteria and on gram-negative bacteria. Now these are the functions of the complement components, and I'm not going to just read it for you because you can just pause it and see what are the different components and what are their immunological functions. But it is good if you can just go through this slide and you check whether what kind of things and what are the components which what they are doing for your examinations. So just I'm just moving forward, and you can just check it. Okay, now let us talk about the complement receptors. So there are four broad structural classes of transmembrane proteins which act as a complement receptors, because complements have to bind on the cells, and on the cells we need receptors where the complement will bind. So we have the four broad structural classes of these transmembrane proteins. First is the complement control protein, which is CCP, or short consensus repeat, which is SCR domain, modular single pass transmembrane receptors. So it is CR1 and CR2. What is single pass? Single pass is when in the membrane the protein goes like it passes through only one time through the membrane. We call it as a single pass transmembrane receptors. The second is the B2 integrins, for example, CR3 and CR4. The third is complement receptor of the immunoglobulin family, which is CRIG. We call it as a CRIG. And then we have the GPCR, which is G protein coupled receptor, which is C5AR1, C5AR2, and C3AR. Now, complement receptors, they act as a bridges between the complement components and the cells to which they bind. Definitely, because if the cells they are expressing the receptors, then the complement will bind to the particular cell, so they act like a bridge. Dendritic cells, follicular dendritic cells, and macrophages, they express all known complement receptors. That means these three cells, dendritic cells, remember dendritic cells, follicular dendritic cells, and macrophages, they express all known four complement receptors. So this is the complement receptors, and again this slide basically summarizes their other name and the ligands, so you can just go through them. I'm not going to read it for you. You can just pause and go through them if you are interested in this. And then it is good if you just know what kind of ligands they are binding to. Now let us see the host defense, how the complement system acts like here. Now, phagocytosis is mediated by many different complement receptors on the surface of macrophages and neutrophils. So here you can see this is C1q, this is MBL mannose. So here you can see this is CBLQ and MBL mannose. Then we have the antigen antibody. So you can see here antigen antibody. We have C3b, C4b. You can see here C3b, C4b. Then we have the cleavage products. So you can see the smaller cleavage products. So phagocytes using their FC receptors. Now phagocytes also have the FC receptors. They bind to the antigen opsonized by antibody binding, right? And the smaller fragments of the C3, C4 and C5 cleavage, which is C3A, C4A and C5A, they act as a anaphylatoxins so that they can induce the inflammation. So anaphylatoxins, and they induce the inflammation. So here you can see these are the different receptors, complement receptors. CR1 is one, CR3, CR4, CRIG, FC receptor, and SIGNR1, SIGNR1. So complement components, they bind to the antigen presenting cells and they boost their phagocytic ability and the secretion of the cytokines. And these components, they strengthen the B cell immune response by increasing the binding strength or avidity. between B cells and complement coated antigens. So basically avidity is what? When the small interactions, they increase the interaction, that is binding strength, increase the binding strength, which is the avidity. So they complement components, they increase the avidity so that stronger binding happens between the antigen and the B cell. Okay, and immature T cells, they are shielded. Immature T cells, they are shielded from natural antibody and complement mediated lysis by extra sialic acid residues on their surface glycoproteins. We have the immature T cells. Now these immature T cells, they are shielded, that means they are protected from the natural antibody and complement mediated lysis, as they have the extra sialic acid residues on their surface glycoproteins. And T cells lacking this protective coating, they are targeted by complement, allowing it to play a quality control role during T cell development. So one of the roles of the complement is also to QC the T cell development. If the extracellular acid residues are absent, then the complement will bind to the T cell and then definitely the T cell is going to die. But in case of extracellular acid residues are present on the T cells, then the complement mediated lysis will not happen. So when C3A, C5A and C3B bind to their receptors on mature T cells, they promote T cell growth, differentiation and survival. So on the mature T cells, C3A, C5A and C3B, when they bind to their receptors on mature T cells, they promote the T cell growth, differentiation and survival. So again, they facilitate the T cell growth, differentiation and survival. Now, when there is an antibody antigen complex, so this immune complexes has to be cleared away. So complement receptors on erythrocytes. So erythrocytes are the RBCs. So they also have the complement receptors. They serve to bind immune complexes because we have a lot of RBCs, and RBCs, they bind to these immunological complexes or immune complexes, and then they are transported to the liver for phagocytosis by macrophages. So here you can see in the blood, these are the RBCs. These are the soluble immune complexes, and upon the complement activation, because the complement C3b, it has bound to these antibodies, and and these RBCs have the CR1 receptor. So this CR1 receptor, it binds to this complement which are already bound to these antigen antibody complexes. So because erythrocytes have fewer receptors than macrophages, the latter can strip the complexes from the erythrocytes as they pass through the liver or spleen. So as they are entering the liver and spleen, so these complexes are snatched away by these phagocytes, and then that's how these RBCs are used to clear the immune complexes. So opsonization provides critical protection against viral infection, and in the contraction phase of the immune response, surplus lymphocytes generated are removed through apoptosis. So these are the two things which you should understand, that when opsonization is there, it provides a critical protection against viral infection, because viral cells are opsonized by these complements, and these complements, they kill the viral cells, as well as in the contraction phase, when the immunity is, it has done its job and now it is about to just retreat, then this immune response surplus lymphocytes which are generated, now they have to be killed, and they are removed through apoptosis. Okay. Let us discuss the microbial complement evasion strategies. There are some viruses which synthesize proteins and glycoproteins that specifically bind the Fc regions of antibodies, and they prevent the complement binding. So basically they don't allow the complements to bind on themselves. While there are some viruses, they also produce proteins that enhance the clearance of antibody antigen complexes from the surface of virus infected cells. So that means even the antigen antibody complexes are there, they are cleared so that the virus can easily replicate there. Some make proteins that induce rapid internalization of viral protein antibody complexes. In some cases, these complexes are internalized, so the cells will never know that this cell is infected. In some species of cystosoma and trypanosoma, they form a protein which disrupts the interaction between C2A and C4B to block the generation of the classical pathway C3 convertase. So now this is the strategy of schistoma and trypanosoma. In case of Pseudomonas aeruginosa, it can degrade C1q and C3C3b using elastase and alkaline protease. So they have the enzymes which can degrade these complement components. Two proteases, SCPA and SCPB, they are derived from streptococcal bacteria. They attack the anaphylatoxin C5A. So these proteases, they cleave or they basically degrade the C5A, which is the anaphylatoxin and secreted by streptococcal bacteria. Fungi, Aspergillus fumigatus, it secretes an alkaline protease ALP1. It is an alkaline protease which is capable of cleaving C3, C4, C5, C1q and IgG. So you can see, these viruses and bacteria and fungus, they are very smart and they want to evade at any cost, and so they have developed so many mechanisms to survive themselves. Now we discuss the regulation of the complement activity. So we have the decay acceleration activity. We have the proteins like DAF, CD55, CRI, and their function is to disrupt C3 and C5 convertases because ultimately they have to be degraded. Then we have the cofactor activity of factor I. So we have the proteins here MCP, CR1 and factor H and C4BP involved, and here the function is to help factor 1 inactivate C3B and C4. And then we have the inhibition of MAC formation. So we have proteins like CD59, which is protactin, vitronactin, clusterin, and they prevent insertion of C5b9 MAC into the host membrane. So no more membrane attack complex formed. And then regulation of the anaphylatoxins. So we have the enzymes like carboxypeptidase NNB, and the function is to inactivate C3A and C5A to reduce the inflammation. We also control the alternative pathway by protein factor H, and function is to bind host cells, competes with factor B and aids factor C1 inhibitor, which is C1INH. It inhibits the C1R and C1S in classical pathway and MASPs in lectin pathway. So these are some of the regulation, how the complement activity is regulated inside our body. Although much is needed, there are more which will become or unfolded later. What if complement deficiencies happen? So if there is a homozygous deficiency in any of the early components of the classical pathway, homozygous means both alleles are absent, C1q, C1r, C1s, C4 and C2, it causes immune complex disease such as SLE, glomerulonephritis and vasculitis. These are immune complex diseases. Individuals may also suffer from recurrent infections by pyogenic, which is the pus forming bacteria such as streptococci and staphylococci. A deficiency in MBL, which is the first component of the lectin pathway, it results in serious pyrogenic infections in babies and children. People with C3 deficiencies, they display the most severe clinical manifestations with recurrent bacterial infections and may also present with immune complex diseases. So C3 deficiency is, it is the most severe clinical manifestations which is seen. C4 deficiency causes more frequent infections with bacteria such as S. pneumoniae, Haemophilus influenzae and meningitis. And C5 to C9 deficiency, it causes meningococcal infections. So you can see, the different complements deficiencies cause different diseases, and particularly, the more we are not able to, our body is not able to control the different type of infections. Now let us see, the omnipresence of innate immunity. You can see the innate immunity is present across all the taxonomic group. It is present in higher plants, it is present in invertebrate animals, in porifera, annelids, arthropoda, and in vertebrate animals, of course, it is present, through from jawless fishes to mammals. While adaptive immunity is absent in invertebrate animals and in higher plants, but it is present in vertebrate animals. Enzymes and enzyme cascades are present in higher plants, but porifera and annelida till now we have not found yet. In case of arthropods and further, yes, they are all present. Phagocytosis is absent in plants while it is present in all the animals, lower invertebrates as well as the vertebrates. Antimicrobial peptides are present through all the plants as well as in the all group of animals. So this is also omnipresent. And then if we see the PRRs also omnipresent. They are present in all the groups. So innate immunity, antimicrobial peptides, PRRs, antimicrobial peptides and PRRs, they are the part of the innate immunity. If innate immunity is present, then definitely these are present. While adaptive immunity, phagocytosis is a part of innate, but then it is not present in plants, but it is present in all animals. But enzymes, they are question marks in porifera and annelids. They might be present, but probably we have not got the evidences yet. So that's why we have put the question mark here. So this slide is, it tells about that innate immunity is present in each and every life form, in higher plants, in invertebrate animals, in invertebrate animals. Now, when we say each and every life form, that also includes, bacteria, that also includes, the fungi and other, but they have some sort of, innate immunity. They have some sort of, protection which is always there. For example, in bacteria, if we see, then bacteriophages, they infect, and then we have the restriction modification system which cleaves the incoming viral DNA, right? In case of fungi also, we have so many mechanisms which prevent the infection. So innate immunity is present everywhere, but adaptive is the hallmark of vertebrates only, right? Now let us see, in plants, if we see the plasma membrane, it expresses PRRs with LRR domain, and this PRRs with LRR domain, it is equivalent to animal toll-like receptors, and these recognize MAMPs, which is membrane associated molecular patterns. Some PRRs, they bind to DAMs, which is the damage associated molecular patterns, and usually formed by pathogens fragmenting cell wall components, because these are the plants, so cell wall components are fragmented. Some pathogens inject toxins into plants, and these are recognized by LRR R proteins. So you can see here we have the R proteins, okay, which have both LRR and nucleotide binding domain. And the primary innate responses are reactive oxygen species and reactive nitrogen species, increase in pH, antimicrobials like defensins, and the enzyme chitinases to digest the fungi wall, or alpha 1,3 glucanase to chew the bacterial wall. Okay. Plants also produce phytoalexins which have the antibiotic activity. So you can see, plants also have the innate system, and very well placed innate system where they can easily protect themselves from the bacteria, the fungus, and the different type of through different type of mechanisms. Okay. All right. So they also have the PAMP PRR binding. It induces the guard cells, which is the stomata, and they close it so that further invasion is prevented. So in this way, we have different small mechanisms which prevent the plant cells, right? So let us just do the final check here, and sometimes what happens that the isolation of infected cells is done by strengthening the walls of the healthy cells around the necrotic tissue or the necrosis. So when healthy cells, they are surrounding the infected cells, then these healthy cells, they usually strengthen their cell walls, their cell walls, they thicken their cell walls so that the infection cannot go inside that. And that's how they strengthen themselves and try to protect from the neighboring infection. So these are some of the mechanisms which you have seen, and for the innate immunity in plants. And let us just close our discussion today here by summarizing what we have learned so far. So we learned about the inflammatory response, then we discussed about ILCs and natural killer cells. We saw the regulation and evasion of innate and inflammatory response, then interactions between the innate and adaptive immune systems. We discussed complements. We discussed the ubiquity of innate immunity, and in one pager we also described the innate immunity in plants. All right. So in this class we have completed the discussion about the innate immunity, and these two classes are important in terms, and they are slightly bigger also in discussion. That's why we have split into two classes. But innate immunity, as you have seen, they are the part of each and every organism, and that's why it's very important to discuss in detail. So thank you very much for your attention, and take care. We will meet you in next class. Okay, till then, goodbye.